Phages are easily enumerated in a sample using something called a plaque assay. So if we use a solution known as our phage stock, which is simply a sample that contains bacteriophages. So this is our 10 to the 0 dilution, and it's our concentrated phage stock. We can serial dilute this into buffer. You can see it getting significantly lighter, indicating the dilutions that are occurring. And so now we have these phages in these bottles that are at a 10 in serial tenfold dilutions. These samples can now be mixed with indicator bacterial host cells in loose molten agar. So we have a culture here of indicator host cells. These will be cells that are infected by the phages that are in one of the dilutions that we've made. So we're going to mix a set volume of our dilution with a set number of our indicator host cells, and we're going to add them to tubes containing molten top agar. You can see those mixes occurring. Okay, so now the cells and the phage will interact together and they'll set up an infection. And because the top agar is still molten, we can then pour it on top of a nutrient agar plate. And if we incubate those plates overnight at 37 degrees centigrade, we'll see an area on the plate where the cells have failed to grow. So just like our culture here is red, where the cells have grown on the top of the plate, we see red growth. And where the phages have infected cells, we see areas where cells have failed to grow. And that indicates where the cells have been infected by these phages and failed to grow in the top agar on the plates. Each one of these areas is a plaque, and it indicates where an infected phage from these infection mixes landed on the top agar. By counting these spots, these plaques on these plates, and multiplying this number by our dilution factor, we can work out how many phages were in our original stock solution or our original sample.